All right, let's get into the DNA structure. So as far as the DNA molecule goes, it looks pretty much like a ladder. And so basically, the DNA is made up of a building block called a nucleotide. And a nucleotide is this guy that's squared off right here. And he has three different parts to him. He has a base, a sugar, and a phosphate group. And so what you'll notice here in this DNA picture is that there are the sides of the ladder, which makes up the backbone of the DNA molecule. And the backbone is made up of those alternating sugar and phosphate molecules. On the inside of the ladder, so the rungs of the ladder, you'll notice these different letters, A's, T's, G's, and C's. So these guys, these are base pairs. And so what's important about these base pairs is that they have a partner that they always bond with. So for example, A's always bond with T's and G's always bond with C's. Adenine bonds with thymine, guanine binds with cytosine. So kind of an easy way to remember the base pairings is apples in trees, cars in the garage. So A's with T's, G's with C's. Another way you can easily remember which ones go together is that the straight-lined letters go together and the curved letters go together. That's another way to remember the base pairings. And these base pairs are bonded together by what's known as a hydrogen bond. So let's talk about DNA replication. Now that we know a little bit about the structure, we are going to talk about the process of how DNA creates a copy of itself. And so that's what this word replication means, is to make a copy. And so with DNA replication, we end up producing two DNA daughter molecules from that single parent. So basically the steps with DNA replication are pretty easy. The DNA has to unwind first. So the DNA is really tightly bound together. And so what we have to do is we have to unwind the DNA so that we have access to the base pairs on the inside of the ladder. So the DNA is going to unwind, it's going to unzip, and now I have all of these exposed base pairs, the A's, T's, G's, and C's, that are looking for a new partner. And so what's going to happen is free-floating nucleotides are the building blocks of DNA, they are going to come floating in and then match up with their complementary base pair. So again, my A's always bond with T's and my G's always bond with C's. And so what will happen is, as these nucleotides are bonding together with their complementary base, we're essentially creating two new DNA molecules from the single one that we started with. Each one of these new daughter molecules, you'll notice in this picture they have a light-colored strand and a dark-colored strand. So another important trait about DNA replication is that both of the daughter molecules end up having one of the old strand from their parent and one new strand that's created from those free-floating nucleotides, and that's called semi-conservative.